Yeah, thanks for the introduction. I work at the NCT, and our group recently grew a lot. So we split the group into two halves, and I get to lead the soft tissue navigation or computer-assisted navigation group. And that means we try to build surgical navigation systems for the surgeon, with a long-term goal of also building navigation systems for robots. So as everyone in the field, we have the problem of data acquisition. And we are actually situated in the hospital. So we have a very close access to the surgical staff, and we record lots and lots of videos. And of course, the problem is always labeling this data. And so we have ongoing projects that deal with this. For example, this is a recent publication where we have 13,000 images, a recent publication from colleagues of mine. And then we have various projects which try to get the labeling and the data acquisition closer to the surgeon. So here, a colleague of mine is building an app where we try to get live annotation on the phone directly from within the surgery, because usually we record these 8-hour-long videos and then nobody looks at them again. And the only way to really process them is through unsupervised work. And we're trying to fix that. But all of this is very different in my field. In my field, in computer-assisted navigation, we have a lot less data than what the other group has. So usually what we need for navigation is intraoperative sensor data on the left, and then preoperative data, which we try to fuse for the navigation system. So we need matching data for a certain patient in order to do that. And usually we have 10 to 20 patients. We might get a few more, but that's about it, where we have that matching data. And in order to validate the whole system, we have usually around 0 patients, because we don't have a displacement field for the liver. We don't know how exactly it's displaced within the surgery. And that is why we need alternatives, and we tend to go into simulation. And we've recently built a lot of simulation tools in the past few years. Lots of them are open source. I think that's sort of why I'm here. And we try to build sophisticated tools that hopefully lots of researchers in our field can use in order to build systems similar to this, or navigation systems similar to what we do. But the first question is, what do we want to simulate? So this is a work from a few years ago from a student of mine who simulated using SOFA, and I think this was in Gazebo, so it's all ROS. Simulated a simple simulation or deformation scene of the liver. And of course, we would like to publish everything here, like the displacement field, the camera images, and so on. And we use real patient geometry here. And the problem is that it scales really, really badly. So building the simulation was basically a whole master thesis. After we had done it, we could probably port it to new patients within hours or days, but getting a huge data set with hundreds of patients is not really feasible in that way. And so we try to take a different route, and we instead simulate randomized scenes that represent those things from the surgery that we need, and randomize the rest. So we start not with, and this kind of makes our approach unique, I think we start not with real liver meshes or real organs, but instead we use these random organ-like meshes that we generate. Then we try to generalize lots of parameters in the scene. So for example, the elasticity is something we take from literature. So each of these organs is assigned an elasticity that is similar to something that a real liver would have, but is not derived from a certain patient. We try to automate the whole process of setting up the scene and building everything. And then in the end, we can parallelize this, and usually we have an order of 10 to 100,000 scenes. And this is also an important aspect at the bottom. We again and again we notice that we can build really sophisticated deep models of that do the downstream tasks that we want to look at or that we want to perform. We spend weeks optimizing that, and we get marginal improvements, and then we scale from 10,000 to 100,000 scenes, and suddenly we have a big jump in improvement. So that's really, yeah, something that we keep on noticing throughout various projects. So our goal is to build a relatively flexible pipeline for various applications in navigation. So we split the functionality into what we call pipeline blocks. These are similar to VTK filters, for example. We've heard VTK a few times. We've heard VTK a few times here. But I think they're a bit more general because, for example, the random scene block would call Blender internally. So we call separate third-party software components and then stream the results back into our system. Each scene is basically created by configuring which which pipeline blocks you want for the scene, and then running all of these pipeline blocks on the scene. And that's what we call a pipeline in the end. And now what's important here is that each of these scenes is completely independent. So we can create lots of them by parallelizing really simply and just running each data sample through each pipeline block. We use another open source tool here called Parcel. We've just switched to this, so this is really new. And it's a really nice library that basically handles all of the pushing to our cluster for us, handles caching. That's a really important aspect when you're trying to simulate 100,000 scenes with soft tissue deformation, and you always get issues like halfway through. And if you don't have caching, then you have to rerun everything. So Parcel, we spent so much time trying to fix that ourselves, and now Parcel basically handles this for us by checking which pipeline blocks still need to run for a certain data sample. Yeah, and these are some of the examples of pipeline blocks that we already have now. I'm not going to go into too much detail on each of them. I'll show some examples of what we can do with this. But the important takeaway at the moment is that we focus a lot on generating the kind of data that we would have in an operation without using actual patient geometry. So for example, we have a block that adds noise to a point cloud, or we have the rigid displacement block, which simulates that before the operation, we usually have the intraoperative data and the preoperative data in entirely separate coordinate systems. So that's what the rigid displacement does. And here's some sample scenes. So the brown thing here would be I took away the abdominal wall so you can look into the scene, but the brown thing would be the liver-like object. We have some ligaments, small pieces up there. We have some fat that the organ is embedded in, and they look very random, and you can see that they're very far from an actual laparoscopic scene. And so what can we do with this? This is part of my PhD thesis, where we took this kind of pipeline, an older version, but similar setup, and simulated preoperative and intraoperative meshes of an organ-like object. So we have some deformation going on between there and between those two objects, and the intraoperative scene is a partial view as you could see through an endoscope. So usually that's below 30% of the surface that you can see there. And the preoperative mesh is a volume mesh. That's really important because that's what we're interested in at the end. We don't really care about the surface. We care about where the tumor and the vessels are that are inside this organ. Then we have various, we call them volume to surface networks, because they register the volume to the surface, which learn to, with our synthetic data, learn to output a displacement field. So they estimate how the preoperative mesh would have to be displaced or deformed in order to match that intraoperative piece of software. Of course, it's an estimation task because it's not well specified really. You could have lots of solutions that fit that small intraoperative piece, right? And we've also done, I'll show that in a second. Here's an example. The white mesh is a real liver mesh, and the yellow is the real intraoperative sparse point cloud that we, not sparse point, but small point cloud that we have gotten from the operation. And then the network estimates the displaced red mesh for each point inside the volume. And the cool thing is that this translates to patient data without ever having seen a real liver. So in this case, we really never, we trained only in the synthetic data. Yeah, and now, of course, we have the displacement field, and we can now interpolate that onto the tumor or the vessels, so that we can then displace those as well. Here's an example of how we apply this. This is phantom data. We have, actually, you probably recognize, some of you probably recognize Orbis. This is all implemented in ROS, and we used OrbSlam, I think 2 it was at the time, to capture more of the liver surface. That's sort of our bottleneck in this example here. This is simulating the fat and the ligament. And now in green, you can see the registration, which is performed by the network, and which estimates, which then is used to estimate the position of the tumor inside the mesh. Sorry, inside the liver. And here you can see the overlay with vessel overlay. When you do this kind of thing, of course, it's really important to know what to validate. And we take several approaches. So one thing that we do is we evaluate during simulation. And when we notice that things aren't realistic enough anymore, so for example, if we have too many intersections after the deformation that can happen due to the random nature of the setup, then we abort that sample and just move on to the next usually. Because we have so many samples, we can just do that. Another thing that we recently started doing is we started adding vessel trees into the simulation as well, because we want to simulate some ultrasound as well. And here we compare distributions of what we generate to the distribution of real patient data. And then, of course, we have these downstream tasks where we evaluate here, for example, how a liver mesh that is, in this case, it has been deformed by breathing motion. So this is the same patient, two meshes, two captures of the same liver with different phases of the contrast agent. That's why the vessels look so different, right? The contrast agent has moved on within the vessels. And the human has breathed between these two meshes, or between these two captures. And so there's a misalignment, and our network is shown the surface and then aligns the internal displacement, aligns the internal structures. So we focus a lot on these downstream tasks and evaluating on those. Of course, the downstream tasks can still fail. So a lot of times we get situations, this is true for everything from simple segmentation of the endoscopic video to these registration tasks. A lot of times we get feedback from the surgeons that they see an error, and it's really frustrating for them to not be able to interact with the system, not be able to fix the problem. And I guess it's generally true that, not trying to be political here, but discrimination is usually easier than creation or generating something. So what we want to do is we want to leverage the surgeon's knowledge and then find ways to incorporate that into the system, either through continuous learning or active learning, or by using the information that the surgeon provides directly on the fly. So here's an example, again, intraoperative surface is generated from the laparoscopic video, or the point cloud is generated from the laparoscopic video, and the preoperative mesh is first roughly aligned here. You can see here that there's a big tumor sitting on top of the surface, and this would be the ridge line. So here's the ridge line, and this is the tumor sitting underneath the surface here. Sorry, not underneath the surface, but sitting on the preoperative mesh, you can see here. And now if we apply the V2SNet, this would be the result. You can see that it improves over the initial registration, but a lot of times we get these errors where the ridge line is not perfectly aligned, for example. And that happens due to various reasons, and I think the main reason is that depth reconstruction is still really, really difficult, getting these intraoperative point clouds in a real scene. Some of you are nodding, I guess. That's an ongoing topic, and so we have lots of noise in these intraoperative reconstructions. And this misalignment, it's due to various issues. This is something that we want to fix, and that's something that the surgeon sees immediately. And what we can do now is that we can ask the surgeon to set various cues on the surface with a very simple tool where they can just use their mouse like a paintbrush, and then set cues that should be matching. So green should be matching green, and blue should be matching blue. And they're rough. They're not precise. It's not an exact match, but this area should be here roughly. And then we input that to something we call QNet, which is an iteration on this V2SNet. And QNet then takes this into consideration to hopefully correct the result. That's something we're trying to publish right now. I think the biggest problem here is that for a lot of samples, the V2SNet actually performs quite good already, and so QNet doesn't show a big improvement. That's been our main issue with trying to publish this. But in this sample, it does improve a lot. So far, I've been talking about the 3D data. Now, there's a trend in navigation to use more of the 2D data and perform 2D, 3D registration, which I think makes a lot of sense because, as I said, it's really hard to simulate to reconstruct point clouds. So this is something we're looking at, putting cameras into our scenes and then also capturing all that data that the camera could see. So here would be on the left would be the scene of a simulated ligament pulling on a simulated organ mesh. Then of course we have segmentation map, normal map, depth maps, and so on. We can get all of that from the simulation. But the problem is that this data does not look realistic. So putting that into any kind of algorithm that performs segmentation, for example, or liver registration is going to fail as soon as we apply it to real meshes. So we've been working a lot on image-to-image translation, and Danush is currently working on a system where he can use liver masks or organ masks and then generate more realistic looking scenes, right? If you look closely, they are definitely not realistic. But the important part is that the region that is masked usually looks a lot like the real liver. So this region looks a lot like liver. And that's something that was really difficult to get right, and we're still working on that. But I think he's making some good progress here. Here's a newer example where he's trying to speed up the process. He's using stable diffusion and control net, and that tends to be quite slow. So generating data for 100,000 patients or scenes is really difficult here, and that's why he's trying to speed that up a lot. And I think he's getting there. So now he's using a simulated scene, and translating that in multiple steps into something that looks a lot more realistic. A difficulty we keep on facing is that realism and diversity and semantics consistency tend to disagree with each other. So if you take this as input and you tell the system it has to keep semantic consistency, so only use liver textures on the liver and only use fat textures on fat, then you will never get these smooth transitions, for example, that you often have in real surgery or real images. So that's something that is really difficult. We have a few parameters that we can tune, and if you turn up too much, then you get really realistic images that no longer correspond to the input masks that you gave the system. And another thing that's unsolved at the moment is we can do this on these scenes where we base the scene on real laparoscopic geometries. So here we used real liver meshes and tried to mimic a real situation. And as soon as we switch to these random organ-like meshes, at the moment the system breaks down, because of course it's trained on real scenes, so it knows what a real scene should look like, and it can't keep the semantic consistency and make it look like a real scene. So that's something we're still working on, but I think we're getting there. I think 3 years ago, another colleague looked at semantic consistency, because so far in this image translation setup, there's no reason for the system to make 2 images of the same patient look like they are from the same patient, right? And if they are trained and translated individually, then if you create a video of those, you get lots of flickering throughout the scene, because there's no consistency over time between these between points in the 3D mesh. So here we spent a lot of time changing the system so that it was able to store information on the 3D surface rather than only in 2D space. And the result is something that is already a lot more consistent. So here you can see that there are regions on the liver surface or on the ligament, for example, which are definitely kept consistent over time. So those are some things we're trying to improve on. One question we keep on posing ourselves: when is it real enough? So in some of the earlier images, you might have seen that, for example, some tools were suddenly appearing where they shouldn't be, or tissues were overlapping in regions where they shouldn't overlap, or fat tissue would leak into liver tissue, for example. And there are lots of scores that we can try to use to find out which image is more realistic than another one. Many of them are trained on natural images, so a bit difficult to translate to our domain, but it works. But the problem is this is not really interesting for us, right? We use these measures while generating data in order to see improvement. But in the end, what we're really interested in is enhancing our machine learning algorithms. So that's a big focus of us. We always demonstrate that using the synthetic data improves some sort of downstream task. A few years ago, I was at Micha, and there were lots of image translation works where they just showed that the images look more realistic. But as long as you don't demonstrate that it works on real patient data, in the end, I think there's not really a point to it. I mean, it's interesting from a methods perspective, of course, but it's I think it always needs to be evaluated on real patient data in the end. Yeah. How much time do I have? Still have 5 minutes. So now that we have the simulation pipeline set up, we can use it for various tasks. One thing that we recently did in 2022 was we participated in a P2ILF challenge. I already mentioned that there's a trend to focus on 2D, 3D registration rather than 3D. So that was the first challenge that I know of that went in that direction for liver registration. So the input to the system was just an image and the liver. And the goal was to register the two. And we were given some labels for the 3D mesh and for the 2D mesh, labels of various regions. And the challenge was twofold. It was really difficult. The first challenge was to capture these or generate these labels automatically, given the data that we had. And then the second challenge was to use these labels to register the meshes, the mesh to the image. And what we did was we used our pipeline to and put these meshes with those labels into the pipeline, and then simulated lots of deformations and displacements in order to generate more data. And so we used our pipeline basically as a really expensive augmentation method. And we did that because we weren't allowed to use any additional data. So we couldn't use our pipeline to generate additional data, because that would have been unfair to the other teams according to the rules. And so instead what we did was we used it to augment the data that we were given. And then the setup that I think all teams ended up using differentiable rendering to try and solve this. And here's an example where we used differentiable rendering to align these labels. And then, of course, if you have the labels aligned, you can also align, or you can use the same transformation on the liver mesh. We won this challenge, but I don't think because we were good from a methods point of view. It was probably more because we used a bit of a brute force. There was no time limit in how long it was allowed to calculate. So we used a bit of a brute force approach and tried lots of things automatically, and then automatically chose the result which, according to these labels, gave the best fit. So our system in the end was slower than others, but produced better results. One aspect that we're always focusing on is translation into the operating room. So you might have seen that even though we're using simulation, we're always trying to focus on getting situations or scenes or using data that is very close to what you could actually see in the operating room. And here are two examples that we're using. We're not using our pipeline directly to simulate data for this, but for the system on the left, for example, that's also using 2D, 3D registration. And we use our synthetic data to parameterize those, or we use our synthetic data to parameterize the algorithms here. So this is an example where a colleague of mine is trying to register rigidly the prostate for navigation, and there isn't a lot of movement going on in this scene, but what's happening is that there are a few places marked in the 2D image, and the 3D organ is registered to these positions, and then they're tracked over time, and it's re-registered every frame, similar to the slide before, what we did for the liver. And then something that we're also focusing on is the actual translation to the OR with taking the medical or the regulation into account. So here we're building a system, the first in our group, that is supposed to help in navigation by, for now, just segmenting structures in the video. And we're putting this onto a medical grade PC, which is which fulfills all the regulations so that we can actually use it in the operating room and show the results to the surgeons live during the operation, and actually feed the image back into the DaVinci, into the Tile Pro. Here we're currently looking at all of the regulations that we have to go through, and we've been documenting for a year now. And the medical product is not going to help the surgery. I'm going to be that open because it's segmenting structures that the surgeons see anyway. But the idea is to build experience and find out what hoops we have to jump through in order to get this into the operating room. So I've learned a lot in that regard here today as well, and look forward to taking that home. We have a few open questions still in our group, so there are things that we're looking into that we can't answer yet. For example, there's some bias in our simulated data. A lot of times you tend, with these random scenes, you tend to get samples where you have very little deformation, and only few samples with a lot of deformation, just because of the way things are set up and because of the reason that, because scenes with lots of deformations are also those which tend to not converge, or which tend to explode because the force was too large, for example. So numerical issues happen more often in those, I think. And at the moment, the best approach that we have is to simulate more data and then choose or sample the scenes non-uniformly, but instead sample those scenes more often which have higher deformation because they occur less often. There's a domain gap, of course, which is simulated in real data. As I mentioned before, we're trying to get realistic, but we're not all the way there, and we're also not necessarily sure we have to get all the way there, and we can't really answer whether at what point we need to stop simulating more realistic data or clean the data that we capture from the actual operating room. The simulation needs to get more realistic. We don't have a lot of tool-tissue interaction in there yet, and yeah, it's also very difficult to know how much we should put into these simulations because iterating is really slow. So every time you change your simulation, you have to regenerate 100,000 samples, which takes days usually, and then retrain your model, which takes days in the worst case, and then you test the model, and then like a week later, you know whether the change that you made has brought any improvement. So that's a real big bottleneck at the moment. And another thing we're looking into is how to combine synthetic with real data. So it makes sense to pre-train on synthetic data and then fine-tune on real data. But we've also we also have experience with some settings where it's really difficult to say how much real data you should use with what learning rates in order to not forget what you learned with the synthetic data. Yeah, sorry, this is an example of a typical depth map that we get, where you can see that there's a ton of noise in this, and that makes it really difficult to work with this kind of data. I thought for fun it would be nice to show what kind of open source tools we use for this pipeline, not just for the pipeline, but in general, that we tend to use almost exclusively open source tools in our lab. And the examples are all these Office tools. Sorry, the counter examples are all these Office tools. I was really glad that I got to present with Google Slides here, because I don't really use Microsoft PowerPoint, and then LibreOffice is just horrible still. So I'm happy to learn about alternatives there as well. Yeah, that's it from my side. The current version of the pipeline is not open source yet, but we'll definitely do that as soon as we progress a bit further.